All right, so my job is to get through the clinical trials section and give us some time to talk a little bit about and to do your question and answer. So what we'll probably end up doing for some of the Q&A, because I don't know that we'll be able to get to all of it, is I suspect if you write down your questions and give them to Sarah, she can direct them to the right speaker to really address your question, and we can try and get you comments back electronically. Michael's saying that that works. So if you've got paper in front of you, if we don't get to your question, please write them down and we'll try and make sure we get them addressed as we go forward. So what I'm going to do is talk a little bit about clinical trials, and I'm going to do some basic 101 about clinical trials, and then I'm going to end with what I like to call some of the really good stories from clinical trials, because I think there can be some amazing things that we see through clinical trial development as we go forward. So what is the purpose of clinical trials in terms of multiple myeloma? And a lot of this can be generalized to cancer therapy in general. We're trying to figure out what new potential treatments are for patients and how to make those standard in terms of how we use drugs and in combinations. One of the key things that patients often ask me is, am I going to get a placebo? And we've got patients here that are on clinical trials, some of whom are on randomized phase 3 clinical trials. And it's important to realize that in both arms of a randomized phase 3 trial, patients are usually getting a drug, a treatment approach. It's whether or not you add the new drug in that's really the question. So there's only one trial that we're running, and I'm going to show you our portfolio at the very end. There's only one trial where we're running where patients either get treatment or they get nothing. And that situation is a trial of smoldering myeloma where the standard treatment is nothing. And so you're getting what you would normally get as part of the trial in the control arm there. So every other trial has active treatment as part of the control group there. Now, when we think about survival in multiple myeloma, you heard David talk about the improvements in survival just in the last decade. It's more than just doubled. And what we quote patients who have standard risk myeloma is that we expect that patients will be beyond 10 years. And that's usually not good enough for many of our patients. And so we say with every new drug that we get approved, we expect to add another year to 2 years on average to the median overall survival. And so there are many patients who are 10, 15 plus years out from their transplant in ongoing remission. And in fact, I'm going to disagree with Dr. Nuka, where in his section he said patients are not cured. I think there are patients that are cured, and I'm going to show you some data that I think speaks to that point. In general, Dr. Nuka is right about everything, but this one point I'm going to disagree with him on. And again, there are many new drugs that are being studied in clinical trials, and they're helping us to understand the biology and concepts of how to combine drugs together as we go forward. So I'm putting this figure up just to show you how many targets we have in myeloma to go after. So every place you see white letters or you see a pathway or something described in both the tumor cell and in the stroma. This is the stroma, and the stroma are the supporting cells in the bone marrow that actually feed the myeloma cell. Every one of these things that you see in black or in white in terms of text represent a potential target of treatment in myeloma. And so when you ask me, why do I need to go on a trial? It's because we don't know which of these things to really go after. We don't know which ones are going to be the best. We don't know whether some things are going to be good for everybody or whether some things are going to be better suited for certain types of patients as we go forward. So this confusion is meant to confuse you so that you now know how we feel when we think about how to treat myeloma. We make it look easy. I think we may try to make it look straightforward, but there's a lot of data that we're trying to put together, and trials are one way that we begin to sort some of that data out. So what are some of the important research questions that are going on? Well, you heard Dr. Vij give a very nice description of a lot of the genetic and genomic information that we have going forward. I just wrote a review for a large trial published from the UK myeloma group where they sequenced, I think it was 600 patients as part of their trial, used what I consider old-fashioned myeloma therapy. And so the net result is that all we know about is sequencing for old-fashioned myeloma therapy. It doesn't really help us make decisions now in 2014. A lot of the work that Dr. Vij is doing is looking at more modern myeloma therapy and using that information with sequencing to help guide how we're going to use that information going forward. So it's not just the technology, it's what are the tools that you're using to treat the myeloma that may influence your outcomes in addition to the information you get from sequencing. What is the best treatment for newly diagnosed untreated myeloma? This is a huge question that's going on right now. And as Dr. Vidge mentioned, we're going to come to we are at 3 drug regimens now. We're probably going to come to 4 drug regimens with the availability of monoclonal antibodies. And then the question becomes not only how good are they, but how are we going to pay for them? Because that is a big issue as you start to answer or add in 3 approved non-generic drugs as part of the standard treatment for patients with myeloma. So these are important questions. And then what are the best drugs or combinations for patients in the relapsed refractory setting? You heard allusions to it earlier that our center is big believers in combination therapy. We think that 1 + 1 does equal 5 when you put the right drugs together. That has to be demonstrated in large randomized phase 3 trials. Otherwise, you're going to end up just combining drugs and not have anything left later on when you need them. So this is actually a snapshot of data from the COMPASS trial. So you have to pretend that you're going to forget this when you walk out of the room, because this is actually the first 400 patients that were sequenced in the COMPASS trial around the world. So these are patients from all over. And what it shows you is a list of some of the more common mutations down here. And one of them, BRAF right here, was actually a mutation that was identified in the original MMRF sequencing paper. And this is actually an important mutation, not from a prognostic implications, because in fact there is now data suggesting that whether you're BRAF positive or BRAF negative, your overall survival doesn't appear to be any different at all. However, if you're BRAF positive, this gives you one more potential tool that you can use to be treated with that you wouldn't have if you were BRAF negative. And in fact, we've got a lady, a very young lady, who's got very aggressive, resistant, high-risk myeloma. We'd run through pretty much everything that we've got, both on and off clinical trials. We sequenced her. She was BRAF positive, and she's now responded. So these can be real lifesavers often, especially if you don't have a lot of other treatment options, and it's worth getting this information at some point to understand whether these can give us clues in terms of targets. As Dr. Vidge mentioned, cancer is cancer. It doesn't matter whether it comes from the lung or the liver or the colon or anywhere else. And in fact, you'll see some of the more common mutations that we see are RAS mutations. RAS mutations occur in almost all diseases, in almost all cancers. So if there's a drug that eventually works for a RAS mutation, you can bet that we're going to bring it into myeloma, because that's a very common mutation that we see in patients with myeloma as well. But mutations are not the whole story. And this is a really nice figure that Larry Boyes from our program actually put together in a review he published a little while ago called The Tau of Myeloma. And Larry's concept, his principal concept, is that targeting myeloma is not just about targeting mutations and targeting cancer. It's about targeting things that make a normal plasma cell a normal plasma cell. And that's things like making lots of antibody. You heard earlier, that's what plasma cells do. Well, our most effective drugs that we have in myeloma, Velcade, Revlimid, carfilzomib, and pomalidomide, all target normal plasma cells. That's how they work. In fact, if all you did was focus on the genetic alterations of myeloma and say we're only going to use drugs based on mutations, you would have missed Velcade, Revlimid, carfilzomib, and pomalidomide. We never would have even started to look at those drugs. So it's not just about mutations. It's about trying to marry picking mutations along with this basic normal plasma cell biology that ultimately leads to success in the context of effective treatment for myeloma. So, misconceptions about clinical trials. Let's go through a couple of these real quickly. The participation gives direct benefit to me, and I may get a sugar pill. I think we've already talked about that. There are no placebos. Everybody gets a standard treatment. It is true that there may be no direct benefit to you. That is absolutely true. We hope that's not the case, but it's certainly possible that there will be no direct benefit to you. I will be treated like a guinea pig. The ability to do trials in patients is so regulated, you would not even believe it. You would not imagine the level of oversight that goes into patients who are on trials. And what I often tell patients, and my clinical trials team, part of them are down here in the front row, is that you actually have more eyes watching you if you're on a trial than if you're not on a trial. We have many patients who have been part or are parts of trials here right now today. I'm sure they'd be welcome to talk to you about their experiences, and I hope they'll say good things. I can't guarantee that they will, but I hope that they will. And clinical trials are for patients with no treatment options. That is absolutely incorrect. And I'm going to show you our menu as we get to the end. Kenesha, who works as part of our clinical trials group, puts this menu together every week, and it gets sent out to all the investigators. We vie over who gets the trial of the week. I've been left out lately, and I'm starting to feel a little underappreciated, but I'm going to lobby to make one of my trials the trial of the week again. But this is how we think about it. We really do think about trials in every situation that we have as we go forward. So how do we get new drugs? This is an important question because many of you are saying, you've got these great ideas. You showed me all the targets. Why don't we have trials for every single one of those? Well, the first thing you have to do is identify the target. And that's what I showed you on that first slide. The target is which of those letters are we going to start to go after. The second thing is you got to find the drug to go after it. And of all those mutations that I showed you on that list that we've seen in COMPASS so far, 3 of them actually have drugs to go after them. Most of the mutations that we see, not just in myeloma, but in cancer in general, don't have drugs to go after those mutations. So it's great to find the target. Then you have to find the drug that goes after the target and do the right studies to prove it goes after the target. And then you do the clinical trials to prove whether that drug actually makes a difference in patients. And ultimately, that's what we care about. And many of you heard me say this before, we are not mouse doctors because mice don't get myeloma. All we really care about is the patients who have myeloma. So ultimately, that's the main thing that drives us in our delivery of effective therapy for patients. What does not the model predict, but what do patients do when they get these new drugs and new targets? So what kinds of trials are there? There are really 3 basic categories of trials. There's phase 1, phase 2, and phase 3. And historically, phase 1 has meant that the likelihood of benefit for a patient is low. Historically, it's been less than 5% of patients will benefit from participating in a phase 1 trial. That is not the case in myeloma in 2015. In fact, some of our best responses have come in phase 1s where we've combined drugs together. So that is a bit of historical information. It's not always the case in phase 1 trials. Phase 2 means you know that the drug is safe. You've figured out that you can give it without bad adverse events. Now you want to know if it works. And so you treat 20, 30, 50, sometimes 100 patients, all with the same dose, and you see whether or not the drug works. Phase 3 is you know it works and you know it's safe. Now you're comparing it to a standard treatment to see whether patients stay in remission longer or whether they live longer. That's really the principle behind a phase 3. So these are the main trials that we do. At Winship, I'll tell you, most of our trials tend to be phase 1 and phase 2. We do sometimes have phase 3, but phase 3 trials tend to be done more in a community oncology setting, not necessarily in an academic setting. So, let's skip one here. What about other types of trials? Well, the reason I think that this is in here is longitudinal studies is basically what the COMPASS trial is. A longitudinal study means you just follow along and see what happens over time. You don't actually intervene. You don't actually dictate treatment. You're only looking to see what happens to patients over a period of time. And this is really important to help us understand what the natural history of myeloma is, for instance. Whether it's treated or untreated, you get that information through a longitudinal study. And finally, the expanded access, which I think are probably what everybody's excited about. It's before a drug is actually approved by the FDA, but it looks like it's going to be approved by the FDA, so the company makes it available for patients across the country. And expanded access, for instance, is how we're going to get access to daratumumab, hopefully very, very soon, for patients who are not on a clinical trial or for elotuzumab as well. So what are some of the drugs that are coming down the pike in the near future in phase 3 trials? Well, elotuzumab is one that you guys have heard a little bit about already. I'm going to show you some data on elotuzumab in a minute. Ixazomib is the oral proteasome inhibitor. You heard it described earlier as oral Velcade. That is actually in phase 3 trials. That will probably be coming down the pike very soon as well. Daratumumab is another a monoclonal antibody that I'm going to show you some information on in just a few moments. And then again, Dr. Rudman touched on denosumab or Xgeva. That's in phase 3 studies as well. There are a couple of other that are in phase 3 development, but these are probably the furthest along of the drugs that we're looking at right now. So what are some of the drugs that we have in phase 2? This is a long list of things, and I think I'm going to show you long lists because I want you to be hopeful when you walk out of here. The reason there's a long list is there are lots of questions being asked in multiple myeloma. And if only one of those trials on each page that I show you hits, that's one more new drug, and that's great. That's exactly what we want. We want as many things to mess around with as we can. So the BTK inhibitor, this is what's actually approved in lymphoma and now in Waldenstrom's macroglobulinemia. The data in myeloma is not great as a single agent, but it's being combined with other drugs. So we'll see whether this drug actually has legs here. CDK inhibitors are something that are based on some of the biology we talk about. A drug we've had a lot of experience with, and I know Jeff is running a trial on right now, is a KSP inhibitor called filanosib or Array 520. There are many other drugs that you can see all the way down the list. Selinexor is another sort of, we hope, breakthrough drug coming in the near future. The preliminary data looks very good. Our trial will open up in the next few weeks here, and I think most of my colleagues have this trial open as well. So these are drugs that we're all very excited about and we'll have access to very, very soon in the near future. What about other drugs in early phase 1, phase 2 trials? Ricolinostat is another one. This is a second generation HDAC inhibitor. It's a cousin of Feradac that they talked about earlier, only it appears to have a much better safety profile than ricolinostat or than panobinostat that we saw earlier. There are many, many other drugs in development. Again, an IGF inhibitor that's in trials in the Myeloma Consortium. OSI 906 is actually open at our site and a couple other sites in the US. And then new and second and third generation proteasome inhibitors like aprocimib and marizimib. Those are both currently open within the Multiple Myeloma Research Consortium as well and represent active potential treatment options in the near future as well. The monoclonals we're going to get to, and just for the sake of time, I'm going to switch the gears to the immune system-based approaches as we go forward. There are a number of ways we can begin to harness the immune system to fight myeloma. And so obviously there's passive immunity called monoclonals, and then there's active immunity with the so-called CAR T cells or vaccines. And I want to talk a little bit about the real difference between these three because fundamentally there are some differences. The first is monoclonal antibodies. And again, this is passive, and this is taking a protein and targeting a receptor on the surface of the tumor cell itself. So in my view, that represents true targeted therapy. It's taking an antibody and going directly after the tumor with a specific target. The nice part about this is that there's no middleman here. You get the antibody, it sticks to the target, and then we hope that the immune system comes in behind and kills it. When you start to think about things like dendritic cell vaccines or peptide vaccines, the hard part is it's like connecting flights. And I use this analogy whenever I'm not in Atlanta because as we know in Atlanta, we don't ever have to connect, right? We go everywhere direct on Delta. But most of our colleagues have to connect when they go somewhere, right? So the connection is like that first flight from wherever city they're in that comes to Atlanta. That's the vaccine. The second flight is the flight that leaves Atlanta to go where they want to go. And that's dependent on whether the plane is there. And if the immune system is not ready for that vaccine, it's like you showed up at Hartsfield and the plane's not there to take you somewhere. And so you are dependent on that second plane to get you where you want to be. The third set is what I consider delivering cells, cellular therapy. Allotransplant that Dr. Wiesel talked about. He's not here, so I can tell you the truth about allogeneic transplant. CAR T cells are another way to deliver cellular therapy. And the reality of both of these are, while they may certainly be effective, you're unleashing the immune system without a set of brakes sometimes. And that does potentially put you at risk for lots and lots of side effects. Doesn't mean it doesn't work. It just means you have to understand the risk-benefit ratio of that therapy as you go forward. So what are some of the potential antibody targets we have in myeloma? Well, you can see some of them down here. The ones in blue are targets that we already have. The two I'm going to focus the most on are SLAMF7, which targets elotuzumab, and CD38, which is right here, which we're going to talk a little bit about daratumumab. But you can see all of these in green are in potential targets, and these in orange are laboratory-based targets, and there are now antibodies being developed against each of those. So this is the list right here of potential treatment monoclonal antibodies that we could have in the near future. It's not where we are today, but these are things that we could get very soon in the near future. So let's start off talking about daratumumab. Daratumumab is an antibody that targets CD38, and CD38 is present on all myeloma cells. So it's a great target in that sense. It's there when you need it to get there. What is the data for daratumumab as a single agent? Well, this is called a waterfall plot. And anytime you see a bar below the line here, that means that a patient's myeloma shrunk. the lower it goes, the more it shrunk. So you can see there were patients who achieved close to 100% reduction in their myeloma on a phase 1 study. So remember when I told you earlier that the historical data on phase 1s said that 5% of patients benefited? That doesn't look like 5% to me here. This is what happens when you treat patients who are resistant or refractory myeloma with a monoclonal antibody early on. As you can see, a small number of patients didn't respond, but a majority of patients did respond over the course of treatment with that monoclonal. Now, if one drug is good, I told you that I'm a combination guy. If one drug is good, what if you combine them? And this is data combining daratumumab with Revdex, lenalidomide and dexamethasone. This is the phase 1 experience over here on the left. Everybody responded in the phase 1, 100% response rate. If you then look at the phase 2 expansion, almost everybody responded. So again, this is what the power of combining drugs together, combining drugs that work through immune activation, like Revlimid and pomalidomide and thalidomide, with monoclonal antibodies. And as Dr. Rudman alluded to, I will be presenting data on the largest trial with daratumumab in 2 weeks at ASCO. Stay tuned. The data looks pretty reasonable. That's all I'm going to say. What about elotuzumab, the other monoclonal antibody that's coming down the pike? This antibody actually does 2 things. It does directly target the myeloma cell. As you can see here, elotuzumab is an antibody, sticks to the target, SLAMF7. But elotuzumab also goes and sticks to natural killer cells. And natural killer cells are cells whose job is to kill cancer. That's their job. They go after cancer cells because that's what they're really primed and effective at doing. So when elotuzumab binds an NK cell, a natural killer cell, it actually turns it on and says, go do your job. That cell may be sleeping or quiescent or not doing anything. Elotuzumab sticks to it, it wakes up, and it goes after the target. And so elotuzumab not only targets the myeloma cell, it also seems to target the immune cells that are then needed to kill the cancer cell down the road. And in phase 1 trials, when we combined elotuzumab with lenalidomide or Revlimid and Dex, we did that phase 1 here along with Ravi and a couple of other sites. You can see 92% of patients actually responded at the right dose, 10 mg per kg. And in fact, if you look at remission duration, it was actually an average remission duration of about 25 months. That means patients stayed in remission for about 2 years. If you look at Revdex alone, it's somewhere around 11 to 12 months. So it basically doubled it compared to historical cohorts. Now, what everybody wants to see is this, and it's there. I just can't show it to you. But it will be available in 2 weeks. And this is the progression-free survival for patients who got Revlimid and Dex in one arm versus Revlimid, Dex, and elotuzumab in the other arm. The abstract is available currently in text, and I can tell you it's a positive trial. There was an improvement in how long patients stayed in remission, but that's pretty much all I can tell you right now. But stay tuned. On my wife's birthday, I'm actually presenting the data. So what about antibodies combined with lenalidomide or Revlimid and dexamethasone? So this is an anti-CD138 monoclonal. This is one CD38, the SAR compound. This is daratumumab, another CD38, and this is elotuzumab, all with lenalidomide and dexamethasone. And look at what you see, 78%, 58%, 87%, 84%. This is the power of the immune system being combined with Revlimid or with pomalidomide. I haven't shown you pomalidomide data because that data is coming out in the next 6 to 12 months as well. But both drugs really seem to work well together with the antibodies, with the immune targeted approaches. So what are some therapeutic vaccines? I talked about vaccines earlier. There are a number of vaccine trials that are going on right now. Dr. Nuka led a vaccine trial for patients with smoldering myeloma at our site that we enrolled on very nicely. We gave the vaccine alone. And then, of course, I'm a combination guy. What do? We combined it with lenalidomide. And we know that at least it looked like it was tolerated pretty well. We're waiting on the data in terms of enhancing immune anti-tumor effects and other things along those lines. But certainly a very promising strategy from the research perspective. So, clinical trials in multiple myeloma. Clinical trials have advanced the care we have in myeloma together. No one receives a placebo. Everyone who is eligible should participate. And I want to show you a couple of quick vignettes here. So this is where Dr. Rudman said we were in the late 1980s. Is that a fair statement? Early 1990s? All right, so median survival was 2.5 to 3 years. This is the most recent analysis from the Mayo Clinic, looking back at cohorts. And if you look at median survival, things began to change here somewhere around the 1995 to 2000 cohort is when the curve finally started to get better. 2000 to 2005 looks even better. 2005 to 2010 continues to get better. And just imagine what 2010 to 2015 is going to look like, because now we have all these great new drugs and we're combining them together. So we have clearly made a big difference from here to where we are here. This is actually our data. This is Dr. Nuka's data on 256. It's now how many patients? 400? More than 400 patients who've all got RVD. Some had a transplant, some did not have a transplant. This is the overall survival for our cohort of patients with a median follow-up of 4 years? 3.5 years. 3.5 years. See, that's how good he is. He knows all this stuff off the top of his head. This is where we are today. So this is certainly steps forward in terms of the progress we're making in myeloma. And when I said that I think we are curing patients with myeloma, this is actually data from Spain. This is before Velcade. This is before Revlimid. This was even before thalidomide. And these were patients in Spain who all got what I would call terrible induction therapy. They all had a transplant, and then they got placebo for maintenance therapy. They got interferon. And if you look here, you can see there is a plateau on the curve of patients who achieved a complete remission at about 20%, who were still in ongoing, unmaintained remission. And here, there's a plateau on the curve of survival 15 years after. About a third of patients are still in ongoing complete remission. So we are curing patients now. We are doing it now. The question is, how do we increase the fraction of patients that we cure from this low number here up to where we want it, which is ultimately everybody? And the way we do that is clinical trials. This is a patient who came to us from Pittsburgh, young lady in her 40s, had been treated up at Pittsburgh, had received bortezomib, Velcade, Revlimid, carfilzomib, pomalidomide, had one transplant, and basically the doctor in Pittsburgh said, I don't really have anything left for you. I'm not sure what else you're going to do. She came down to us. We looked at the genetics of her bone marrow, identified that there was one drug that might actually be very effective for her based on work from Dr. Boyes in our laboratory. It's a drug that targets something called BCL2. There is a small subset of myeloma patients who are exquisitely sensitive to BCL2 inhibition. We put her on that drug, no Dex, no Velcade, no Revlimid, just the BCL2 inhibitor on a trial. This is what her myeloma protein did over the course of 6 weeks. This is what her myeloma protein has done over the last 18 months. She now flies back and forth from Pittsburgh every 3 weeks in ongoing complete remission because of a clinical trial. Second story. I've got lots of these. I'm only going to show you two. Second story. A patient who came to us from the outside, who'd had multiple treatments over the course of years, had seen everything that I just talked to you about before, had low counts, didn't really have a lot of great options in terms of treatment, and came to us because they wanted to get on an antibody trial. This patient has been on this antibody trial for over 18 months, and again, you can see what happened to his light chain over the course of one cycle, quickly went into a complete remission, and has been there for 18 months. One drug, monoclonal antibody, and that's it. These occurred because of clinical trials. So when you ask me, why do I always talk about clinical trials, every drug up here that any of you in the audience have ever been able to use came through our program, came through the collective programs of my colleagues, and we wouldn't have them today if people before you all hadn't gone on these trials to demonstrate their benefits. So it really does make a huge difference. It does make a huge difference. This is the menu that Kenesha and our program puts together every Monday. We get this list every Monday, and in fact, it's a PowerPoint slide, and if you click on one of these links here, the eligibility criteria come up. It's the coolest thing. I mean, I said it one day, and Kenesha did it like a week later. I thought, wow, I was just making it up. I didn't think we could actually do it. But as you can see here, we've got lots and lots of trials for patients in every single situation and state. And it may not apply to you, and we may tell you, don't go on a clinical trial because we don't think it's the right thing for you to do. But if it is the right thing to do, or it gives you something different, you can see it's broken down by newly diagnosed. We have trials for Waldenstrom's. We have observational trials like COMPASS. Patients with bad kidneys, we have special trials for early relapse, late relapse, early phase 1 trials with new drugs and new targets. Lots of stuff across the board. This is what you need if you need some new treatment. So I think it's a great option. And with that, I will stop, and I think we'll bring the panel up for questions.